Welcome to Learn Biology, and we're still on the topic of organic chemistry, but today we're discussing a huge topic and it covers proteins. So let's begin. What are proteins made out of? Well, proteins are made out of the four basic elements: carbon, hydrogen, oxygen, nitrogen, and also sulfur. Now, proteins are polymers, and they're made out of amino acids, which will abbreviate AA throughout the video. And amino acids are monomers. But what proteins are used for in our bodies and mammals and in almost every other organism out there is for muscle growth, gene expression. This is the big one. The structure on our face, what our eyes shape looks like, almond shape, wide, how our cells are designed. All this is related to proteins and genes. Reactions that take place within our cells, energy release and making of polymers and destroying polymers and making monomers and so on. So reactions. And an example of a protein, a pure protein, is albumin. Now what is albumin? Well, it is the egg white. So here's an egg, and the inside, the egg white, is albumin. Alright, so as we said, proteins are made out of amino acids. That's a very big concept right there, amino acids. And there's some basic things we need to know about amino acids. Proteins are simply chains of amino acids. Here's an amino acid. It is linked to another amino acid. And this link in between, the thing that holds the whole amino acid together, this right here is called a peptide bond. And it is a type of covalent bond, again called peptide, right here, peptide bond. And what peptide bonds do, it's again a type of covalent bond. If you need review on what covalent bonds are, please check out my other videos. But proteins are linked together by amino acids, and amino acids are held together with peptide bonds. And now here are some basic terms: dipeptide, tripeptide, and polypeptide. Di means two, tri means three, and poly means many. So when there are more than two amino acids hooked together, and there are when there are two peptide bonds, so here's one peptide bond, here's another, and here's an amino acid. This right here is known as a dipeptide bond. Now let's say we have again this, another amino acid, this, and then another one. So 1, 2, 3. That's tripeptide. And polypeptide are more than 3, and usually they average at about 50 peptide bonds. 50 peptide bonds. That's a big number, but that's the average size of a true protein. 50 peptide bonds. Alright, so the structure of proteins. Well, there are three main structures of proteins. There is primary, secondary, tertiary, and then large proteins are quaternary. So what primary is just a chain of amino acids. Right here is a primary structure, and all it is a chain of amino acids, a string of amino acids. So just AA hooked to another AA, and so on. Now this is the initial stage, so the name primary. We go on to secondary. Now secondary is when the amino acids seen here, the string of amino acids start to coil up. They form, so they right here, as we can see, a coil is formed. So this is what a secondary protein structure is, where the amino acids coil up because hydrogen bonds occur in between. So here, for example, we have an amino acid hooked to another amino acid. Now what might happen is at an interval there might be a hydrogen bond, and this is why the secondary structure coils up. But all that you need to remember is that secondary structures are coiled strings of amino acids. Tertiary structures are when the coils start to fold on itself. So here, let's say we have a coil. Well, what happens to this coil is that it coils onto itself, so right over and over, and just becomes chaotic. That's what a tertiary structure is. And it begins to coil up on itself because of the large string of amino acids, along with interactions with other reactions and groups, and these coil up because of these things called R groups. We move on to quaternary. Quaternary is basically just it's links of tertiary structures, so links of these hooked together. So quaternary is the final structure. This is where it becomes a true protein, final structure, and it's just a complex of two or more chains of amino acids hooked onto each other, folded, and then connected. So for example, and there are three different types of quaternary structures. So for example, we'll start with this one. This structure here has 4 chains of amino acids coiled up and connected. So 1, 2, 3, and 4. Now this right here is called a tetramer. Tetramer structure. And why this is because it has 4. Now the order goes is 2 chains is called a dimer, 3 chains of amino acids coiled up and connected is called a trimer, and 4 is called a tetramer. So thank you for watching Learn Biology, and see you next time. We'll be finishing the proteins.